Welcome to the third part of the video about the cell. This video is good for AQA, Edexcel, and OCR. Now let's continue the topic. Let's talk about the plant cells. Plant cells are eukaryotic cells, which means they share many structures with animal cells. These are the nucleus, the cytoplasm, the cell membrane, and many other parts. But plant cells also have additional structures that animal cells don't, like a cell wall, the chloroplast, and permanent vacuole. These exostructures are linked to their autotrophic lifestyle, and that's why the plant can prepare its own food using the sunlight energy. So keep in mind, plant cell include all the structures of animal cell plus three important structures as well. Now let's talk about the extra structures in plant cells which are present in addition to the structures in animal cells. The first one is cell wall, and it's made up of cellulose. What is the function of cell wall? Its function is to provide the strength and support to the body. Also, it prevents the bursting of the cell. The second important organelle, that is the chloroplast, and the chloroplast consists of chlorophyll, and the process of photosynthesis occur because of the chlorophyll. The third structure is vacuole. It consists of the cell sap, and its function is to control the turgor pressure inside the vacuole, inside the cell, and it also store the different substances. Keep in mind that plant cells have all the structures that are in animal cell, but additionally three structures are the cell wall, the chloroplast, and permanent vacuole. Now let's talk about the structure of the cell wall. So the cell wall is made up of cellulose, which is a structural polysaccharide. Cellulose is formed from long chains of glucose molecules linked by beta-1,4 glycosidic bond. The fibers are arranged in the crisscross pattern, making the structure extremely strong. And because of this design, the plant cell is tough, supportive, and polypermeable that allow the water and most substances to pass through easily. The next is the function of the cell wall. So the cell wall plays several important roles in the plant cell. First, it provides structural support, allowing plants to stand upright. Second, it prevents the cell from the bursting when water enters. The cell wall gives rigidity to the plant. That's why the plant can stand upright. Now it's chloroplast. Chloroplast contain chlorophyll, which is found in the thylakoid membranes. A very important point to remember is that chloroplasts are only found in the plant cells, not in the animal cells. Also, root cells don't have chloroplast because they are underground and don't receive the light. This is the very common exam question, so make sure you remember it. Now let's talk about the structure of the chloroplast. Chloroplasts are disc-shaped organelles surrounded by double membrane, and inside it contains the thylakoid. The thylakoid are membrane-bound discs stacked together to form structures called the grana. The fluid surrounding these structures is called the stroma. Chloroplasts also contain chlorophyll, which absorbs red and blue light for photosynthesis. Like mitochondria, they have their own DNA and ribosomes. One thing that you should keep in mind that the chloroplasts are found in the green parts of the plant, like the leaves, the stems, but it's absent in the roots. The next is the function of chloroplast. So the main function of chloroplast is photosynthesis. In this process, carbon dioxide and water are converted into glucose and oxygen using light energy. And in leaf cells, especially the palisade mesophyll cells, there can be around 50 to 100 chloroplasts in a single cell. This shows us that how important the process of photosynthesis is. Now the permanent vacuole. This is the third unique plant cell structure in the permanent vacuole. The permanent vacuole is a large central fluid-filled sac that occupies most of the volume of the plant cell. It is filled with cell sap. As I told you earlier, that cell sap is a solution of water, sugar, and salt, and sometimes the pigments. The vacuole is surrounded by a membrane called the tonoplast. What is the function of permanent vacuole? So the permanent vacuole has several important functions. The first is it maintains the turgor pressure. This keeps the cell firm and turgid. You know the herbaceous plants? Those plants that are without the woody tissues, they rely entirely on turgor pressure for the support. Now the second function is that it stores useful substances such as sugars and salts as well as waste products. Pigments stored in the vacuole also give flowers and fruits their color. When a plant loses water, the vacuole shrinks, the cell loses turgor, and the plants wilt. This is called flaccid cell. When the water enters the cell by osmosis, the vacuole swells pushes outward against the cell wall, and this will become turgid. So it becomes rigid and firm. Now let's talk about the algal cell. These cells are simple photosynthetic organisms. The examples of these include Chlamydomonas, Spirogyra, and seaweed. They're similar to plant cells in that they have a cell wall, they have a chloroplast, and a vacuole. However, they differ from plants because they have no true roots, stem, or leaf. They have a simple overall structure. We can have a unicellular algae as well as the multicellular algae. Unicellular algae has only one large chloroplast, while the multicellular algae have ribbon-like chloroplast array. Microscopy. As you know that there are two main types of microscope you need to know. One is the light microscope and is also called the optical microscope, which has a resolution of approximately 200 nanometers and can magnify up to 1500 times. Light microscope, you can see structures such as the nucleus, cell wall, chloroplast, and vacuole. When we talk about the electron microscope, we cannot forget about the TEM and the SEM. TEM stands for transmission electron microscope and SEM stands for scanning electron microscope. Overall, the electron microscope has a resolution of approximately 0.1 nanometers and can magnify up to 500,000 times. With the electron microscope, you can see ribosomes, membranes, viruses, and DNA. Let's talk about the SEM and TEM in detail. The SEM can scan the surface of a specimen with the beam of electrons, producing a three-dimensional image of the surface. The TEM fires electrons through a very thin specimen to produce two-dimensional, high-quality, detailed internal image. The TEM has a higher resolution than the SEM. The two terms you must not confuse, and that is resolution and magnification. What is meant by resolution? It's the ability to distinguish 2 points as separate. Higher resolution means finer detail is visible. In case of higher resolution, the two closely packed dots will appear as two distinct objects, not one blurry mess. Magnification is how much bigger the image is compared to the actual object. For example, a magnification of times 400 means the image is 400 times bigger than the real object. You can have high magnification with low resolution. The image will be larger but blurry. Keep in mind that resolution and magnification are not the same thing. To calculate the total magnification of a light microscope, we have to use this formula. Total magnification equals eyepiece power multiplied by objective power. For example, if the eyepiece lens is 400 times and the objective lens is 40 times, then the total magnification is 10 multiplied by 40, which equals to 400 times. Here is how we use a light microscope correctly. So the first step is you have to place the slide on the stage and secure it with the stage clips. The second step, select the lowest power object lens like time 4 to start. The third step is move the stage up from the side, not while looking through the eyepiece, using the coarse focus knob. In the fourth step, look through the eyepiece and use the coarse focus to get rough image. Next, switch to the higher power lens, times 10 times 40 as needed, and then use the fine focus knob to sharpen the image and adjust the light intensity by opening or closing the diaphragm. To prepare a microscope slide, you have to follow these steps. First of all, place a drop of water on a clean glass slide, then place a thin specimen on the slide and lower the cover slip at an angle to avoid creating air bubbles, and then add a stain to increase contrast and make structures visible. Two common stains, iodine, which stains starch blue, black, and is used for plant cells, and methylene blue, which stains nuclei blue